Well, it's my pleasure to be here, and also really fun to get to follow such great speakers this morning. I feel like so much has been covered already, and there's so many really exciting technologies that have been described, like these biosensor microbes, that if we could integrate them together, would provide lots of really exciting insights into soil processes. So I'm at Berkeley Lab, although I'm not sitting at Berkeley Lab. This is what it would look like if I were. This is, you can see the Golden Gate Bridge, and this is up on the hill up above UC Berkeley. And I'm also at the Joint Genome Institute, which has now moved from Walnut Creek back onto the main site. So these are the two topics that I'm going to cover today. The first is going to be focused on kind of setting up the rationale for the need to monitor, understand, and ultimately harness metabolite exchange in the rhizosphere. And I'll go through some science that we've been involved in that really made us feel like we needed tools, sterile ecosystems that we could use to study metabolite exchange in the rhizosphere. And then in the second portion of my talk, I'm going to tell you about efforts from many individuals to build fabricated ecosystems and define microbial communities that can be used to study these processes. So for the first topic, we've already heard a lot today about the importance of the rhizosphere. I think you guys are all very familiar with this, but maybe just to hit a few highlights. Plants deposit a large fraction, they can deposit a large fraction of their fixed carbon as rhizodeposits, including small molecule exudates. This is primary input for plant microbe interactions or for selecting for beneficial microbes. It's thought that these metabolites that are released by the plants are helping structure the rhizosphere communities. And this process is really important to the global carbon cycle, as we've heard about. Plants are a primary input into soils. And also this is, these microbes, these plants are not releasing carbon for no reason. It's thought that this is selecting for microbes that are helping provision the plants with nutrients. And so this makes the rhizosphere a really important environment for sustainable agriculture. So for example, learning how to have support low input production of bioenergy and biomaterials. And as we've heard this morning, these exometabolites that are being released by the roots are selecting for epiphytic microbes that live on the rhizosphere and on the surface of the roots, and also endophytes that come into the root and confer various functional processes on the plant, and that the plant is using both active transport and then also likely some passive transport to produce these metabolites in the rhizosphere. And this is from a fairly recent review paper. I put this long list here just to show you how diverse these exudates can be. And this is just a little portion of the various metabolites that are transported by plant roots. I also wanted to note that this is a two-way process. Some of these metabolites, we typically are thinking about these organics being pumped out by the root to feed the rhizosphere bacteria and fungi, but microbes are also releasing compounds that are uptaken by the plants. And these metabolites can be simple things like organic acids that may mobilize organics from mineral surfaces, as we heard about this morning, or sugars that may feed microbes at the root surface. But there also includes trigolactones and flavonoids, as we also just heard about, that have additional functional benefits in terms of communication. And there's a really diverse range of secondary metabolites, and these are especially poorly understood. So tremendous diversity here. And how can we advance an understanding of the functions of each of these metabolites in selecting rhizosphere bacteria? And this is the way that we think about it in our lab. These exudates are kind of like a buffet, and we're interested in understanding how what you lay out at the buffet affects which microbes are supported in the rhizosphere. And the way we measure this is using what we call Exometabolomics, which is simply performing metabolomic analyses on exogenous metabolites like root exudates. And the technical approach is very simple. We separate the metabolites from whatever environment we're interested in, either using some sort of liquid extraction, something like that. And then we separate them based on their physical properties using HPLC. And then those metabolites go into a tandem mass spectrometer, which separates them based on their mass to charge ratios and allows us to measure many properties of those metabolites, especially the fragmentation pattern and the retention time, that allows us to identify those metabolites and link them to microbial genes, et cetera. And the workflow for studying exudates is very simple. We take the plants, we collect their exudates, and then we individually culture different rhizosphere bacteria on those exudates, and we measure what's left at the end of the process. And by doing this for a number of microbes, we can start to hypothesize what these metabolic webs might look like in the rhizosphere in terms of what the plant's releasing and what the microbes are using, and what the microbes are producing that may benefit the plant host. And yeah, we just mentioned Mary Firestone. Okay, so here's a paper by Mary Firestone, and this was a collaboration with Owen Brody and Mary Firestone that was led by Kate Zalminia. We did this several years ago, where we wanted to examine the rhizosphere effect. And in an earlier study, Mary's group had identified this strong reduction in actinobacteria over a 12-week period when Avena barbata was growing. So the actinobacteria became less abundant and the alpha proteobacteria became more abundant. So a nice example of a rhizosphere effect here. And so what Kate did is she collected exudates at each of these same time points here and did exometabolite profiling. And we were interested, first of all, to see if exudates change over time. And in fact, they do. And that was kind of a fun thing to learn, that early on, the plants are producing a pretty simple mixture of metabolites, mostly just sucrose. So kind of here's some candy for attracting the microbes to the rhizosphere. And then later on at week 6 and 9, we started seeing a more complex mixture of metabolites. And then as the plant started to senesce, we saw betaines, compatible solutes, and hormones and things like that. So then what Kate did is she took collections of these isolates and fed them the exudates and used exometabolite profiling to identify if there were, if some of these metabolites that we detected being produced by the Avena barbata could explain the enrichment of the alpha proteobacteria. And she did find that many of these alpha proteobacteria like to eat the aromatic acids, including phenolic compounds, and that this potentially is a tool that the plant is using to select for the rhizosphere bacteria. And she also found that many of these compounds are actually toxic to some of the other bacteria. So this could be sort of a, both have two mechanisms of selection. So then in a more recent study, we asked, does the plant alter exudate profiles in response to nutrient stress? And this was, again, a collaboration with Mary Firestone, who organized and led this work. This is a very nice study that linked to a field site, and actually Mary's group and others collected soil from the field site, brought it into the greenhouse, packed columns with soil to get the right densities of the different layers. And then this is very low carbon marginal soil. And so then they amended it with different levels of fertilizer to study these nutrient effects, and sampled along the rhizosphere for both microbes and metabolites to look at the role of these aromatic acids and other compounds in the rhizosphere. And I'm just going to quickly go through some kind of highlights here. The first thing is that when you look at the marginal soil versus the nutrient amended, we have more of these aromatic acids present in the rhizosphere during the nutrient stress and also pentosis. And we didn't identify which pentoses these are, but pentoses are more abundant under the nutrient stress. And here's an example of chlorogenic acid, which is one of the compounds we detected in our earlier study. Now, not surprisingly, when you add nitrogen amendments, you start seeing more nitrogen containing compounds. And so this is the increase versus the marginal soil once it's been nitrogen amendment. And you see many amino acids that are more abundant. One thing that caught Kate's eye when she looked at this is serotonin, which is not something that we would typically be looking at in soils. And it had a significant increase once you amended the soil with nitrogen and phosphorus, or just nitrogen versus the marginal soil. And serotonin is a real functional metabolite that's been shown to act as a natural oxidant inhibitor in Arabidopsis thaliana. So we thought, well, this is pretty neat. So some of these metabolites may have functions beyond just being substrates for microbes. And we can also, in addition to looking at the changes in the abundance of the metabolites under these different nutrient amendments, we can do the same for the microbes. And so when you add nitrogen or nitrogen and phosphorus, you see an increase in the actinobacteria and loss of some of the oligotrophic groups like acidobacteria, Planctomyces, alpha proteobacteria. And so now that we had both the abundances of the metabolites and the microbes, we could start looking at correlations between the two of them. And we find that amino acids have a positive correlation. Here's this scale here, and this is a Spearman correlation, are positively correlated with actinobacteria and alpha proteobacteria. Like they like those as substrates. And then we also see a positive correlation between these aromatic acids, which are the ones that we're particularly interested in, and some of these more oligotrophic groups. And once again, serotonin caught our eye as being one of the most strongly correlated, or in this case, anti-correlated and positively correlated metabolites. So this is kind of like a hub metabolite in this study. And so we decided this is so interesting, we wanted to follow up on this. And so what Kate did is she added, we want to see, does it actually do something for switchgrass? I mentioned that there is a study out there in Arabidopsis showing that it has some physiological effects on the Arabidopsis. So she added 100 micromolar serotonin to 7-day-old switchgrass and incubated for 18 days. And wow, we saw a big effect of the serotonin. So, I think this is a nice, example of how these metabolites can have a big impact on the plant host. And this is in terms of the biomass of the shoot biomass here, the root number, secondary root number. So very large fold changes with the addition of this single metabolite. So altogether, we've gained some insights into different classes of metabolites that may have some impact on the plant host. They can be structuring the rhizosphere community. And we really wanted to test these in more detail and under conditions where we could efficiently sample the rhizosphere for microbes and metabolites. We could ideally localize where these processes are happening in the rhizosphere. And importantly, we wanted a sterile ecosystem so that we can control the microbial composition within the rhizosphere. As a first step towards this direction, Kate did a very simple experiment where she made a collection of 1,000 isolates from the same field site that I mentioned earlier in Oklahoma. And she wanted to see if she could basically simulate a rhizosphere effect by taking a soil extract and then adding these aromatic acids that seem to be, we hypothesize are structuring the community. And here we don't have any root or anything like that, which is just taking the isolates from the field and adding a mixture of these compounds with the soil extract. And she found that shikimate and ferulic acid were almost 100% used by this mixture of, I think it was about 100 isolates. So those were their favorite compounds in there. And she saw a bloom of microbes that were, the same kind of microbes, same genera of microbes that she found enriched in her earlier study. So maybe an initial indication that this chemistry really is a strong controlling factor in structuring the rhizosphere community. But again, we didn't have a plant root here. So we really wanted to develop some devices and capabilities to be able to study these processes directly in the rhizosphere. And that leads me to my second topic, which is the need for fabricated ecosystems. So almost everything that we know about biochemistry and molecular and cellular biology has been learned studying model organisms. We also have a vast knowledge about field processes, and the challenge is linking the two together. And so what we see as a necessary bridge between the laboratory and the field are what we're calling fabricated ecosystems. So simple, devices that maybe are closer to a model organism, but you still can start, you can visualize the roots, see how the physiology of the plant is being affected by the microbes. You can collect the exudates, and importantly, you can control the microbial composition. Maybe this is much more like a petri dish, for example, and then developing these fabricated ecosystems that are more representative of the natural field processes, but still contained and controlled, so that, for example, this topic that came up at the end of Carrie's talk, you could still use engineered organisms, these gas reporters, for example, in a system like this, which is what we're calling an ecopod. And then ultimately these link to the field studies so that you can gain new insights into soil carbon cycling and also sustainable bioenergy. We've been involved in a number of workshops hosted on this topic, and just this year we had our first little mini conference on fabricated ecosystems. And one of the workshops yielded what we call the EcoFAB design principles. I'm not going to go through this in detail right now, but I'd be happy to come back at the end of my talk if anybody would like to discuss this. Basically, a large group of diverse scientists working on studying microbe-host interactions came up with a vision for developing standardized community resources that could be used to advance microbiome science. And that's summarized in this paper here, where Karsten was the lead author on. And it shows this vision for a workflow where you create reproducible laboratory habitats, defined microbes, microbiota that are accessible to anybody in the community, say through a tissue culture collection. Use 3D printing to make these devices, so anybody can make them now, given the widespread use of 3D printing. You create defined protocols for how to do the metabolite extraction, the DNA extraction, proteomics, et cetera. Baseline measurements so that people know whether their laboratory ecosystem is behaving to specs, so to speak. And that critically, we have to have the ability to make spatial temporal measurements, not only of just the localization of microbes, but also where metabolites, proteins, we heard great examples of optodes, where oxygen and pH, we really need to have this more complete view of the environment and the microbial communities within the rhizosphere. And once this technical platform has been created, then need to perform interlaboratory comparisons to validate that it's performing the same in multiple different laboratories. And then this becomes a resource for the community where people can read a paper and they can duplicate the results and they can extend it to their own research. These designs should be modifiable so that people can take it in their own directions and really enable determination of mechanistic processes. And another strength of having a standardized community fabricated ecosystem that that are being used to study the rhizosphere is that machine learning can be used across multiple experiments, which is really, typically we don't have enough observations in a given experiment to make machine learning, as useful as it can be. So having many studies that are using the same system will greatly increase the number of observations for machine learning. And I couldn't resist throwing this in here. In terms of extensibility of designs, Kirsten Hofmackel at EMSL has been pioneering a new development in EcoFAB technologies, which he calls the soil EcoFAB, for studying biofilms in soils. And I'm not, it's just a teaser, but I just couldn't resist mentioning that, you know, I'm talking about this Plant-Microbe ecosystem today, but there's also a soil EcoFAB that's been developed. As one of the workshops highlighted the need to have a steering committee to help oversee this and organize ring trials, et cetera. And so we formed a really great steering committee that includes Carrie, who you just heard from, and many others. So coming to, you know, creating this reproducible habitat, when we looked at existing devices, there wasn't really anything that was quite right. And so we decided we needed to create what we call this EcoFAB device. And here were some of the key properties that we wanted for this device. And I'll just highlight that we really wanted a sterile system where these, you know, this system was a little too small for studying the rhizosphere and doing the imaging. These weren't sterile systems, although these are all powerful and excellent technologies. So we thought we needed to build a little smaller device. This was the design that we came up with. So it's just a silicon rubber device on a microscope slide that forms a root chamber. And you have a plant port where the plant grows. And I'll show you some pictures in a second. And the roots grow down along the microscope slide so you can image them. And then you have some sampling ports for collecting the exudates. If you're interested in this, this Jove article goes through all the step-by-steps for making this, and all the designs for making these that I'm going to show you are available on this website here. We decided to use Brachypodium distachyon, which is a model grass and is relevant to essentially all bioenergy crops, or at least most bioenergy crops are grasses, and so this is it's a great model for those. It's also relevant to grain crops, so also very important for food production. And John Vogel, our key collaborator here, literally wrote the book on Brachypodium, and he's standing in front of some very small portion of his mutant collection. So there's a huge collection of mutants that we can use here, including chemiluminescent versions. So it's a very nice model system to use. We worked with Peter Kim and Anup Singh at Sandia National Lab to develop 3D printing capabilities to make molds for casting these devices. Making it is actually really simple. You 3D print a mold, just like making Jell-O, and you pour in the monomer solution, you heat it up, and you remove it like a big sticker, and then you chemically bond it or clamp it onto a microscope slide, and you've got your little chamber, and then you germinate some seedlings and you put them inside the chamber within a sterile box, and you have this metabolite system for studying rhizosphere interactions. This is a very old figure from when we first created this, just showing that we could study this metabolic exchange in the rhizosphere. So what we see here is the plant growing by itself, and here's just a few metabolites that we put down that are accumulating over time as the root is growing. So like glutamine here. And then we inoculate it with different rhizosphere bacteria, and we start seeing the consumption of these metabolites and the production of new metabolites that are, you know, products of the microbes. So we can see sort of the microbial consumption and the plant release, and, you know, just highlight that serotonin is one of these compounds that we detected. So I think this is a really powerful tool for studying the chemical dynamics in the rhizosphere. So moving over, you know, we've got the device, we've got some protocols. Wanted to do a ring trial experiment to see how reproducible this Brachypodium EcoFAB system was across multiple labs. And so we had organized 4 labs. This was all work by Joelle that organized this interlaboratory comparison study, where she created little kits with the EcoFAB devices, seeds, different media, and a sensor to monitor the incubators in each of the laboratories, sent them to the 4 different labs, and then characterized them over 3 weeks in terms of using imaging, Then the samples were collected for analysis of biomass and phosphate, and also root metabolites and exudates, which we profiled at JGI. And so these are the different treatments in terms of the media that was used to grow the plant. And we can see the fresh weight here and the phosphorus content. And so we saw significant treatment effects and similar results between all of the different laboratories. So it showed nice reproducibility between laboratories. And then when we look at the root metabolites and the exudate metabolites, they cluster by treatment, not by laboratory. So this has provided some evidence that this is a reproducible system. Also, the imaging results was dramatic and reproducible across laboratories, where we observed that a soil extract promoted very dramatically longer primary root hairs for some unknown reason. So seemed like a good resource tool in terms of research tool. And then in terms of extending it to other systems, we've collaborated with many other labs to test these devices out for other model organisms or model plants like Ceteria and lotus, sorghum, medicago, switchgrass. They all grow well. And then Ritesh, who used to be in Jerry Tuskin's lab, had to try making a gigantic EcoFAB, and he was able to grow Populus trichocarpa in it. So that was pretty fun to have this gigantic EcoFAB. So the basic design seems to work for many plant hosts. We've also collaborated with a company to test out these devices for different types of food crops, and they seem to work well there for studying different plant interactions and the role of plant microbes in supporting plant growth. We've sent some of these to Jeff Bowman at UCSD, who's using them to grow mangrove, so possible connections to wetland plants like we heard from Zoe this morning. And one problem that we had with these EcoFAB devices is that even though we have a narrow root chamber, the roots go out of plane. So it's very difficult to get a high resolution image along the length of the root. So Lauren and Peter developed what they call this imaging EcoFAB, and here's the mold for it. It has a lot of little fingers that basically allow diffusion of nutrients, but they pin the root against the microscope slide. And the plants grow very well in this device. You can see you can take this device and put it into a little magenta box with a sterile filter, and it makes it very easy to change the media. And they've collaborated with a number of people, including Yasuo Yoshikuni, who's used a synthetic biology tool to make many different colors of bacteria. In this case, these are all Pseudomonas fluorescens with different fluorescent proteins. And they were able to measure these metabolites, measure these fluorescent proteins in the rhizosphere and generate some very pretty images looking along root here. I think these are root hairs, and you can see single bacteria. So this combination of synthetic biology, like Carrie talked about, but in this case for making fluorescent constructs with these EcoFAB devices, I think can really help us learn about the biogeography of rhizosphere microbes. Naji at UC Berkeley has been using these same devices to use really advanced spectroscopic methods like the third harmonic generation or 3-photon imaging, these are both labile-free techniques that allow you to go directly within tissues. So she originally developed this, I think, for doing non-invasive imaging directly within brains, and now she's applied it to roots, and so she can go, for example, image where vacuoles are, and I think this has some potential for imaging endophytes directly within living roots. And Yasuo has also made some chemiluminescent constructs, which are really powerful in that they are easy to image directly within EcoFABs that contain soil, which can make it more challenging for fluorescence imaging. And so here's just some examples of switchgrass growing in soil, sand, and in a hydroponic medium. And then imaging them, we can see that the microbes are, they really like to grow at the root tips in the hydroponic, also in the sand. In the soil, you still see them at the root tips, but they're also growing a little bit more at the plant base here, at the base, possibly due to oxygen limitations in the soil, which again connects back to Zoe's talk this morning. So I think a really important direction to take this, as I mentioned earlier, is to study spatial systems biology. You know, you would not go study a savanna by, you know, extracting everything and losing all that spatial information. It's, you know, the context of where the resources are, where the environmental constraints are, and where the organisms are. They're really critical to studying ecological processes. And so being able to develop spatial omics, I think, is a really important research direction to start. You know, I think I've provided some evidence that these aromatic acids could be selecting for the rhizosphere communities, but, you know, where are they being produced along the root surface? You know, and I, you know, we've shown that the microbes that are enriched in the rhizosphere are elevated versus the bulk soil, but everywhere, or is just in certain spots. So I think there's a really important direction to go in. And, you know, I think these chemiluminescent images show us that there is, you know, definitely microbial preferences for where they're localized. And, you know, likely, you know, you'd think the exudates are probably higher here, or at least the compounds that the microbes, that this particular microbe likes to grow on. So Romy Chakraborty's lab with the talented postdoctoral fellow Moon Yi has been studying the biogeography of soil microbes using these EcoFAB devices. They also just wrote a review paper on devices like this. And the first thing they wanted to do was to benchmark EcoFABs versus other more conventional growth devices, including test tubes and pots. And they did this all in a greenhouse where they moved each of the growth systems around periodically to control for the light variation. And they had versions that were in sterile chambers, and also open to the air, so they could see how important it was to control that environment. And they used soil fresh from a field site for this study. And we were all happy to see that the EcoFAB had similar performance in terms of the root, the dry shoot weight, shoot length, and root length to the other devices. There wasn't really any, you know, significant differences between the open and closed devices, though there were some directional differences. It's also nice to see that the EcoFABs, if anything, were maybe a little bit more reproducible. So in terms of starting to investigate the biogeography, they were able to dissect different regions of the EcoFAB device and other chambers. Here you can see the different chamber types. Here's the different locations that they sampled. So the bulk soil, the base of the plant, where I showed you there was that increased activity right at the base, and then also at the root tips. So they sampled those separately, and excitingly, we saw that these Solorubobacteriales, which are an actinobacteria that we observed to be strongly correlated with amino acids and were fast growers in the rhizosphere, were also enriched at the root tip. And so, you know, starting to be able to connect the biogeography to potentially exudate composition. However, we really need a way to do the spatial exudate analysis to be able to really put these two things together. And so we have made some attempts to do a couple different types of mass spectrometry imaging. In this case, we used a surface-based mass spectrometry, which you can almost think of it as like a solid phase extraction medium. It's a very hydrophobic surface, and we clamped this surface, it's called a NIMS chip, against, we replaced the microscope slide at the bottom of the chamber with this NIMS chip, clamped it against the root, and then as the root is growing, the metabolites that are being released by the plant zorb onto this surface, and then we can open it up remove the NIMS chip, and then do mass spectrometry imaging. We did this. It was, we saw mostly phospholipids, which, is a pretty typical thing to see in a mass spectrometry imaging experiment. We did not see the phenolic acids, which were the compounds we were most interested in. And we've repeated this study more recently using MALDI imaging, where we took a stainless steel target, we coated it with a thin layer of agarose gel, we grew the plant on top of it, and then we can see, again, various ions that correspond to metabolites. It's very difficult to identify what they are, but none of these masses correspond to the phenolic acids that we're interested in. And I would just say that this is something that I think a collaboration or a user project with EMSL would really benefit by, because EMSL has really exceptional capabilities in mass spectrometry imaging. So we found so many different uses for these EcoFAB devices. And this is Jason, who was one of the early people in the lab making these devices. And he could make about 6 to 8 per day. And so we weren't really able to keep up with demand. So we hired some more undergraduate students, and pretty soon they couldn't keep up. And and then we hired more, and this was just not working out. So we decided we needed to just bite the bullet and buy injection molds so we could make tens of thousands of these devices. So this was a really big project, and it was led by Peter Andeer, who's a scientist at LBNL. And this is the device that he's developed. We call it the EcoFAB 2.0. It has a microscope slide at the bottom, and then a root chamber, and it has 2 sampling ports that we put septa over, so we can have gas tight sampling, or we can put covers on them that we can remove easily for exudate sampling, changing the media, etc. Then we have this sterile chamber in here. It's kind of like a petri dish lid sitting on the top. It's screwed together, and then we have gas sampling ports on the top for stable isotope analysis. It's made out of polycarbonate, so it's a nice, has good favorable optic properties, and also is a relatively inert material to use. This device has the footprint of a 96-well plate, so we can do automated liquid sampling and introductions. And the height of it is set by the condenser on an automated microscope that we have. So this can be put right under this automated microscope for high resolution imaging of the rhizosphere. And so we created we took that, I told you about that 1,000 isolate collection from the rhizosphere at this field site that Kate made. She selected down to 16 isolates that span the diversity at the field site to create a standardized microbiome as a community resource. And then Karsten Zangler has been using this CN-on device, which allows you to manipulate individual bacteria to look at the right, the optimal ratio of different microbes, given that they all grow at different growth rates. What's the right ratio of these 16 isolates to get a desired community structure? And he's come up with what we're calling the Zangler ratio, which is 1 fast grower to 100 medium fast growers to 1,000 slow growers for each one of those. And this results in reproducible growth. He's also worked out cryopreservation methods so that we can freeze the community, distribute it to other laboratories, and then they can resuscitate it. And he's got a resuscitation media worked out and get the same community structure. And this is in preparation. And now John Vogel is planning a ring trial with this microbiome with a number of investigators around the world. It'll be 5 different labs. And we're going to look at host selection for these isolates. So there's going to be the 16 members switchgrass community plus P. simiae by Lotus japonicus and Brachypodium. And this is what, these, a rack full of EcoFABs looks like. And one of the challenges now is that to process 50 EcoFABs, for example, takes many, many hours for somebody by hand. And, that increases the chances of error. So Peter Andir, Tom Vess, and LT Kornmusser have been developing what we're calling the EcoBot. So this is a robot to perform these EcoFAB experiments, and it has an environmental chamber on the back that has tunable LED lights so that the light quality and intensity can be set to a field site. And it can vary over the course of a day to mimic, sunrise and sunset. And then all of the EcoFABs are sitting on a heat exchanger so that you can have the temperature of the roots mimic the temperature profile in the field. And it has a liquid handling deck in the front for doing all the sampling and changing the media, and then has a couple different imaging stations. There's an epifluorescent microscope and a hyperspectral camera. And we hope to include other measurements, like optos, like we heard about earlier, would be fantastic. Here's just a couple little videos of it moving around. So here is just grabbing EcoFABs from the environmental chamber over and putting them onto the liquid handling deck. Here it's taking it from the liquid handling deck and moving it down into the different imaging stations. And yeah, so here you can see the EcoFAB being moved by this multi-axis robotic arm, getting set down here. And then there's a pipetting robot that comes over and changes the media. I don't need to, if you guys are interested, I can come back and play more of these videos. And we're very happy to have this EcoPod now at Berkeley Lab. And this is designed to be the next step in the EcoFAB experiments. And I want to draw your attention to a poster by Nancy Hess, and she's going to be talking about this really exciting new DOE-funded project called the Twin Ecosystems Project. This is a collaboration with Carrie and Joff at Rice University that's actually going to use those novel sensors within EcoFABs and EcoPods to study important soil processes. We're also collaborating with CAMRA to develop autonomous experimentation in the EcoBot. And we're going to use these integrated capabilities to study the role of exudates during drought and selecting for beneficial microbiomes. EcoFABs are available through the JGI annual CSP call, so you can get them that way. But my understanding is that they're going to be coming to EMSL soon. So you'll hopefully be able to get those through EMSL directly. So just to quickly summarize, aromatic acid exudates contribute to the assembly of grass rhizosphere communities. We have observed a strong association between rhizosphere bacteria and exometabolites. These EcoFABs enable analysis of the localization of bacteria and metabolites within the rhizosphere. In our initial evidence, we have some initial evidence for the spatial and temporal exudate dynamics in the rhizosphere, but much more work needs to be done there. And we feel like these standardized microbial communities in combination with fabricated ecosystems have the potential to advance microbiome science. I'd like to thank all of the awesome collaborators and people in our group for their contributions to everything I told you about, and all of the funding from the DOE Office of Biological and Environmental Research Genomic Sciences program, which has supported this work. And with that, I will stop, and I would be very happy to answer any questions.
Thank you, Trent. Lots of interesting technologies and tools, and I'm happy to see the future EMSL collaboration opportunity there as well. We have a number of questions. The first one is what proteins act as serotonin receptors in plants? Any similarity with animal systems?
I don't know. I mean, there's only, as far as I know, there's only that single paper on serotonin and Arabidopsis, and I do not remember offhand what receptor is binding to the receptor for serotonin. It's a very interesting question. You know, serotonin obviously a big deal in the gut and in human health, and I think that's what really drove drew our attention to it. You know, you see, whoa, there's serotonin in the soil. Where's that coming from? So exciting stuff to follow up on. Thank you.
Next question is, could the addition of synthetic aromatics and other target exudates potentially be used instead of microbial inoculates?
Yes.
It goes on biofertilizers to target and facilitate long-term presence and function of ideal consortia of PGPB and circumvents some of the issues of using inoculates in the field.
It's a great question, and again, it kind of follows this analogy to the animal systems, right? So could you use prebiotics as a way of, you know, structuring the community, either in combination with your target microbes that you hope to get enriched to give them a selective advantage, or without the microbes as sort of like a biofertilizer? Could you potentially use waste streams that are enriched in these metabolites, or develop fermentation approaches to create phenolic acid rich substrates that you could use in the field? I think that's a really exciting direction. Yeah, thank you.
Next question is about the ring trial. How were metabolites measured? Were they shipped and measured at the lab, or compared measurements from different labs? What are the trade-offs? That's
an insightful question. Yeah, so in metabolomics, there are ring trial experiments just to compare reproducibility between different metabolomics labs. So to avoid that confounding factor, we did all of the metabolite analysis at JGI. So all the samples got sent to us. In turn, you know, each lab did the imaging. They did the root biomass measurements, the root phosphate, you know, the shoot biomass and phosphate. They did those. We felt like those were reproducible enough that they could be done by the individual labs. But yeah, all the metabolomics was done at JGI. And I think this is an opportunity to use these fabricated ecosystems in combination with the user capabilities at EMSL and JGI. Because if each of the users are generating these samples and sending them to EMSL and JGI, then you're going to get consistent data across experiments, right? Because the same analytical methods are being used.
Great, thank you. The next question is about, so whether the plant EcoFAB and soil EcoFAB parts are integrated platform that work together, or if they are individually operated, can they be integrated?
Oh, another great question. We thought about that. The idea that you could have like a plant EcoFAB device and then a soil EcoFAB device, and then metabolites could flow from one into the next, and you could kind of almost like simulate a soil horizon. Maybe you could put some of Carrie's synthetic soils in there. You could change the mineralogy. Nobody has done that yet, but I think that would be a really exciting thing to do.
Great. I was wondering about the technique that you use with EcoFAB for, you showed some root exudates measurements. So when you look at exudates or hotspots, is that using some carbon isotopes?
What we've done so far is just simply, I showed a little bit of mass spec imaging, and then dissection and sampling. So when we do the dissection and sampling, we're just doing LCMS-based analysis. As part of this TWINS project I mentioned, we are going to use C11CO2, which has it's a radioisotope with a very short half-life, and it's used. Boy, we're making lots of analogies to biomedical sciences today. It's used for like localizing tumors and things like that, right? So you can go take an image, you can see where the tumor is. We want to try to use this for hot spots and hot moments in the rhizosphere. Other groups have already done that, so I think this is very, very possible. But we would do it within the EcoFAB devices, where we flood in the C11CO2, collect PET images over time, and then dissect after the number of half-lives, go remove portions of the soil where we saw the activities for the omics analysis. And I think that's really going to start getting at which metabolites are being produced when, and how are they impacting the rhizosphere.
And then what will be the methodology for root imaging in that system?
So in that case, it'll be PET, positron emission tomography imaging. I do think that there's so much potential for the mass spectrometry imaging. That's something that Chris Anderton at PNNL is an expert at, for being able to look potentially at enzymes that directly within the soil system, as well as metabolites. Like I mentioned in our initial work, we didn't detect the metabolites that we're most interested in. So developing, there's also some derivatization chemistries that are used at EMSL. Could you possibly use those to be able to detect the compounds of interest? I think there's a lot of potential there. And then also integrate that with, fluorescent microbes, gas reporter microbes, optodes. It seems like so much that could be integrated.
Okay, great. And we have a comment here in research conducted here with, Kim Hickson's comment, with myself and Stephen Callister here at PNNL on Setaria grown on marginal soils with nutrient addition. We also identified not only significant serotonin increases, but also significant increases in synephrine, right? I'm not sure, another alkaloid. It is possible that many alkaloids containing both nitrogen and carbon sources are important in soil microbe remodeling.
Yeah, wow, that's a really neat observation. I mean, it's cool to hear that you've observed something similar in Setaria, as well as these alkaloids. I mean, I think the whole world of specialized metabolites in the rhizosphere and their, again, their localization, how they're impacting both the plant host and the rhizosphere microbes is pretty uncharted territory. And then also for us to be able to differentiate between the nutritional value of metabolites for microbes versus other functional roles for those molecules. I started off and I showed just a small taste of the diversity of metabolites that are present in the rhizosphere. You have to think that they probably have lots of diverse functions, right? Beyond just this microbe likes to eat this one and another one likes to eat another one. And I think developing the technologies to be able to get at the mechanisms of those metabolites is going to be really important, and that's going to require genetic resources too. And so synthetic biology, as Carrie talked about today, is a really important approach to integrate with these fabricated ecosystems.